Good morning, everybody. So today we are going to discuss about biologics manufacturing. Biologics are basically drugs. In contrast to we know drugs that are available today in the market mainly they are of pharmaceutical small molecule drugs. What do I mean by small molecule? All conventional drugs, as we see, they are basically small molecules. Take for example aspirin, take for example azithromycin. All these drugs are actually small molecule drugs which are conventionally produced. Here we will discuss today biologics drugs. Biologics which are basically non-small molecule drugs and are categorized as large molecule drugs. So this is the drugs that are being developed since 1980s. Company called Amgen, Genentech in the US, they have actually started making these drugs as a pioneer. In India, we started making these drugs probably in the year of 2004, 5, 6. Before that, India was actually developing and marketing small molecule drugs, all the conventional drugs. So why biologics drugs are important for discussion today? Well, these drugs are significantly different from the small molecule drugs in one key aspect. That is, they have a good target. They can deliver targeted delivery. So therefore we get specificity, high level of specificity. Now let us discuss about these biologics drugs and its manufacturing. Why these are significant in today's pharmaceutical world. And we've also analyzed how these drugs are actually getting developed more in number than conventional drugs. So in today's lecture, I will try to discuss what are biologics, timeline for a biologics development journey from discovery lab to the clinic, and then how biologics are different from conventional drugs, and then key stages for antibody therapeutics manufacturing. These drugs, biologics drugs, beside having specificity, high specificity than small molecule drugs, for example, take the case of cancer. Biologics drugs are exceptionally specific. For example, a class of biologic drugs are monoclonal antibody therapeutics. They are exceptionally targeted to the cancer cells. How these targeting is achieved, we will know from today's discussion. And why these drugs are being developed, why it is being now has become a big industry in India, we will know from today's discussion. So let us go to see what are biologics drugs. So biological drugs commonly referred to as biologics, which are a class of drugs that are produced using a living system. In contrast to living systems such as microorganisms or animals, this is a contrast to that conventional drugs, conventional pharmaceutical drugs, which are small molecule drugs, which are also referred to as small molecule drugs. to a chemical synthesis process. This is a key difference between how biologics or biological drugs are manufactured vis-a-vis the small molecule drugs. So they are different from chemically synthesized small molecule drugs. Think of aspirin, in that they are generally larger and more complex molecules. So larger and more complex molecules plus chemically synthesized small molecule. So the key difference is conventional pharmaceutical drugs are basically very small in molecular sizes, whereas biological drugs actually are very large in molecular sizes, and their synthetic route is actually small molecule drugs are normally synthesized to chemical route, whereas large complex molecules which are biological drugs are synthesized through living system. For example, microorganisms and animal cells. So biologics, some other differences of biologics are usually administered by injection or infusion, and most biologics in cancer care are given under the close supervision of a medical professional. Well, it is the administration, not the prescription. Administration of biological drugs cannot be done at home or maybe unsupervised by a without a medical supervision. So that's also a difference of biological drugs vis-a-vis small molecule or conventional small molecule drugs. Now let us see what I was referring, what do I mean by referring that biological drugs are actually large and very complex molecules. Here, this plot, this cartoon actually shows, you see on the left hand side, this is aspirin. The size of aspirin is 180 Dalton. And you compare that, this is one of the most earliest conventional pharmaceutical drug. The size is actually 180 Dalton. And you compare that to all other on the right hand side. Insulin, erythropoietin, monoclonal antibody. These are actually biological drugs. And insulin has a size of around 6 kilodalton, erythropoietin 30 kilodalton, and monoclonal antibody is a 150 kilodalton. So there is a huge difference in molecular sizes if you compare against a small molecule drug like aspirin, such as 180 Dalton versus monoclonal antibody. It is like if aspirin is a pinhead like this, you think about monoclonal antibody as a football. If you compare the sizes, I mean, roughly this will be the comparison: a pinhead versus a football. So, why this molecular size is important, we will know, because it brings complexity to its manufacturing for the large molecules, whereas small molecule drugs would be comparatively easy to manufacture. Now, here this table actually I wanted to show that what are the key differences between small molecule versus large molecule drugs. So small molecule drugs are also called traditional drugs. I have been referring this in this talk. I will be referring like traditional conventional drugs, and when large molecule drugs are normally called as a term with a common term as biopharmaceuticals. These are also called biological drugs, biologics, and in India, which is a copy of the innovator molecule, we also call biosimilars. So biopharmaceuticals is a term which encompasses all of these molecules. Even vaccines are included in biopharmaceuticals. But in this talk, we will be restricting mostly to biologics, the drugs, therapeutics, not vaccines. So the key differences are, in traditional drug, you have an unspecific binding. I just discussed that targeting, specific targeting to the cells where the drug will show to the cell or to the organ, where the drug has to show its efficacy for the treatment of a disease is, in case of biopharmaceutical, it is very highly specific binding. In case of traditional drugs, it is unspecific, untargeted. Now, another difference is interaction with other drugs happens because small molecule drugs is non-specific and it does not actually target the cells or the tissues damaged by the disease or dysregulated by the disease, whereas biopharmaceuticals, these interactions are there. Another reason for this is also that biopharmaceutical drugs are actually mostly human proteins. So they are natural to our body and they have been, although they have been produced outside our body, but these proteins are actually natural to our body. So you are using the same proteins as drugs which we normally have in our body for various functions. Then carcinogenic substances possible in case of traditional drugs. Why? Because small molecule drugs are these chemically synthesized, they are non-natural to human body. So, and their properties are chemicals. They are chemicals, they are not proteins. So, they are chemicals and they can be carcinogenic. It has been proven. In case of large molecule drugs, mostly they are non-carcinogenic. Then pharmacokinetics is difficult in case of traditional drugs, and in biopharmaceuticals, breakdown is predictable for the most part. Pharmacokinetics means how, absorption of the drug, its distribution, and then metabolism and its excretion, ADME. So the pharmacokinetics of the drugs actually determines how the drug will show its efficacy, how long it will stay in the body, and show its therapeutic efficacy. So pharmacokinetics is a property, very key property for drugs, and in case of large molecule, mostly they are broken down as our natural proteins go through this route. It's broken down and then get excreted. The same thing happens for biological drugs also, biologics also. So that is also a difference between these two. Then immune reactions, but this is the key positive thing for traditional drugs where immune reactions are rare. They are chemically synthesized small molecule. So, and these they cannot generate immune reactions. Immune reactions can only be generated by proteins or peptides. So, these immune reactions are very rare in case of traditional drugs, mostly not present. Whereas in biopharmaceuticals, this is a safety issue because it shows immunogenic effects. And if you take proteins multiple times in our body, it will definitely generate immunogenic reactions. And immunogenic reactions are sometimes in case of vaccines, we need higher immunogenicity. But in case of other diseases, we definitely do not need immunogenicity. So it's a safety issue, and this is a very big issue in case of biopharmaceuticals, and we need to take care of this. And in case of traditional drugs, another advantage is that theoretically any target molecule can be reached. Nowadays, you can actually design drugs, small molecule drugs, for any target. And because it is also very not easy, but comparatively easier than large molecules, you can actually synthesize and test that millions and millions of compounds for a particular target. That is not the case of biopharmaceuticals. Biopharmaceuticals or biological drugs are actually they target cells receptors, other soluble receptors, proteins, antigens, which are dysregulated in a disease. So that in case of biopharmaceuticals or biological drugs, these targets are limited. You cannot actually have the number of targets as it is possible in case of traditional drugs, in case of biological drugs. Then in case of traditional drugs, it has been observed that 6% success rate in phases 1 and 3, whereas in biopharmaceuticals, it is 25% success rate in phases 1 and 3. This is also an advantage for biological drugs. And in case of traditional drugs, development cost high, production cost low. Here the development cost low, production cost high. So this production cost, why it is high, that is actually the main discussion point in today's talk. Now, these are the top drugs in 2023 by sales, and the values are given in $1 billion. So we see 5 biological drugs and 4 biological drugs and 1 vaccine listed here. So one is Stelara, it's a MAB, monoclonal antibody therapeutics. 2 is Comirnaty. We all know Comirnaty, it's an mRNA COVID-19 vaccine, and Stelara is used in Crohn's disease and ulcerative colitis. And then 3 is Dupilumab, and that is actually used in the treatment of eczema and asthma. And then we have Humira, of course, which is a blockbuster drug, and its Humira's value is around, its sales value is around $14.5 billion. It is used for rheumatoid arthritis. And then, of course, the crown of all of it is Keytruda, which is used for head and neck squamous cell cancer. And you see the value as $225 billion global sales in just 2023 alone. That tells that the efficacy of these drugs, because this $1 billion actually not directly translated into prescription, but most of it can be directly translated into the value can be translated into prescription. So these drugs are very highly effective drugs, also biological drugs. So this I am showing only to show you that out of say 7 or 8, 5 drugs or vaccines are actually biological in origin. That is the significance of this pharmaceutical, as opposed to conventional traditional drugs, which we know that it sells globally very big. We will know after some time what is the total pharmaceutical value in terms of global sales. But now it is been observed that biological drugs is actually growing at a much faster rate than conventional traditional small molecule drugs. And it is the same story actually gets reflected also. It is reflected in FDA approvals also. You see, this chart shows FDA approvals over the years from 1998 to 2022. And the dark blue lines are actually for small molecule drugs. New molecular entities, NME, they actually define as that in the FDA balance. And in case of biologics, these are the light blue ones. So you see, from 1998 to almost 2016, there is very little biological drugs have been approved by FDA. After that, actually the numbers got increased, and in 2022, we see almost 50% of FDA approved drugs are biologics. This tells a big story that earlier, all these years, we used to have uh, biological drugs, uh, very less approved because its discovery and manufacturing was actually complex, and it was not very easy to bring it to the market. It was not very easy to bring it to the market. But now, from 2017, you see the numbers are actually very has increased, and in 2022, we see it is catching up with the small molecule drugs. And these are some of the biological drugs that are being used, and these are blockbuster drugs, we call it. Keytruda, just mentioned in the last slide. Um, and then Stellar also mentioned, Dupixent, Dupilumab also mentioned, but earlier we have Herceptin, Rituximab, and then of course the first biological drug is insulin, also there, and then filgrastim, which is GCSF, granulocyte colony stimulating factor, which is used as a supplement to chemotherapy. And because when neutropenia happens in cancer treatment because of chemotherapeutic agents, um, small molecule drugs, uh, to increase the neutrophil count, you use uh, filgrastim or pegylated version of GCSF. GCSF or pegylated version, granulocyte colony stimulating factor. So it increases the neutrophil count. So this is Neulasta, then epoetin or erythropoietin. We all know erythropoietin actually helps in erythropoiesis in our body, which actually is being used for treating chronic kidney disease, anemia, all those things, because we know erythropoiesis get affected or dysregulated in kidney disease or anemia. So Epogen is first. These two are actually one of the very first. Besides insulin, these two are the very first two drugs of Amgen, which are still the revenue earners even today, revenue earners for Amgen. Some reports say almost half of the revenue of Amgen is coming from Epogen and erythropoietin investing. That is GCSF. So we now, by this time, we actually know there are biological drugs and they are gaining traction over the years, and now it is almost 50% of the total drugs approved in the US by FDA are biological drugs, and we have seen the complexity in sizes, comparison of sizes between small molecule and large molecule drugs, and we know that there are certain key differences between these traditional drugs and large molecule drugs. Now, let us explore how drug development takes place and what is this timeline. Very shortly, this is an FDA drug approval process. Drug approval process, and we have a basic science research which actually takes around 2 to 5 years. Then we have preclinical testing. You need to test your drug in animal models and show safety, efficacy, PKPD parameters before even going for the human studies. So that timeline is around 1 to 2 years. Then you have 3 phases of clinical trials: phase 1, phase 2, and phase 3. You have, and here is the chunk of the drug development process. The chunk of the timeline of the drug development process takes place around 9 years. So, and then once the clinical trials are successful, you know your drug is very, very highly effective, efficacious, and it is very safe to be used and tolerated in humans. Then you take it to the markets and you go through the FDA approval process. It can range between 6 months to 2 years. So around 12 to 15 years, timeline where you can bring a biological drug from discovery to the patient clinic. And of course, this 12 to 15 years, how much that will cost you? So, if you are working on a cancer, anti-cancer drugs or biological drugs, this is an estimated median expense of new drugs approved by the US FDA. So, you can see it's been published in this article. And if you are working on oncology and the immunology therapeutics area, it is around $2.7 billion. So, it's very costly to bring biological drugs from discovery stage to the clinic. And for other disease areas like anti-infective, gastrointestinal, central nervous system downward, you see around 1.2, and then 700 million and over 700 million for dermatology. So on an average, I mean, you can easily say that biological drugs, if you want to bring to the market, it will cost you from discovery to the clinic, it will cost you around $2 billion. So, not everybody can manufacture and bring these drugs to the market. Only big players who have a lot of money to put into the R&D first over a period of 3, 4, 5 years, 6 years, and then doing preclinical and clinical around 9 to 12 years, they can actually work on this. And as far as molecular complexity, these are the structures, driven structures for the molecules which I have just briefly mentioned earlier, like filgrastim, pegfilgrastim, erythropoietin. Erythropoietin is a long-term, long-lasting version of erythropoietin. Then you have hormones, growth hormones, and sex hormones, actually folliotropin, leukotropion, gonadotropin. And then you have antibody molecules like rituximab, adalimumab, trastuzumab. All these are being, whatever I have mentioned, these are actually protein molecules which are being already developed as drug and are being in the market. So I have taken one of the example to its molecular structure to show you why it is important to understand the complexity of the molecule and complexity of the molecule, which in turn actually significantly impacts its efficacy and safety. Let us take an example of a monoclonal antibody. We have already shown you that there are too many antibodies that are available in the market, and many are in the development. And antibody molecules, besides being used as drugs in disease conditions like ulcerative colitis, rheumatoid arthritis, antibody therapies are mostly developed for cancer treatment. So, let us check with the monoclonal antibody. Take an example of structure of monoclonal antibody. It has two chains, a Y-shaped structure, and the most critical, and this top portion is, we all know, this is the Fab portion and this is the FC portion, and the two arms are actually Fab portion, and here in the bottom stain is an FC structure. In the FC, there is a site for a carbohydrate chain to be attached. This process is, we call it, is a glycosylation process, and this is post-translational modification. It happens in our naturally in our body, and in the drug, biological drugs, if you are using an antibody, it will also have these PTMs, post-translational modifications. Now, why did we study these modifications? Because these modifications, post-translational modifications of the carbohydrate structures, they may have a lot of variants, structural variants. And like this, you have, these are carbohydrate chains, and you see there are a lot of variants. What are these variants? These carbohydrate chains are made mainly N-acetyl glucosamine, mannose, galactose, glucose, and glutamic acid. And two key components for anti-cancer monoclonal antibody therapeutics are actually galactose and fructose. So, content of galactose in the antibody molecule and content of fructose in the antibody molecule are very, very significant for its efficacy. So, data shows, for any antibody molecule which are used as an anti-cancer drug, they show their main mechanism of actions for the treatment of cancer is by two pathways. One is CDC, complement dependent cytotoxicity. Through this pathway, it kills the cancer cell. Another one is antibody dependent cell cytotoxicity. So these two mechanisms of killing cancer cell by the antibody drug depends mostly the presence of galactose, the yellow circles, and fructose, the red triangles. It is very interesting to know. The more the galactose content in an antibody molecule composition, the more you will have CDC activity. That means you will kill more cancer cells. And the lesser you have focus molecules in the antibody molecule, you will have the higher ADCC activity, another mechanism to kill the cancer cells. So here it is inverse relationship, whereas in case of galactose and CDC activity, it's a positive correlation. It is interesting because it is interesting. You see that these two structural features in an antibody molecule could control the overall efficacy of the antibody drug in the treatment of cancer. So we must know the variance, its percentages, and its presence, its content as it is present in the composition, and that content can actually regulate how good this antibody will show its efficacy in cancer therapy. So in this particular example, I have actually given the percentages, relative proportions of these glycovariants present in a composition. I'm not naming the name of the drug where these particular percentages are present. You see the G0 variant, where G0F, where you have galactose is not present, whereas fructose is there. That is the highest. That is one of the highest, 35.5 relative proportion compared to others. And you have G1, G2, galactose 1, you have 2 galactose, you see the yellow circles. Then G2, and then you have sialic acid as well as fructose content. So this way you will have, I have shown maybe most of the variants, glycostructural variants in an antibody molecules. There are many more, and besides affecting the efficacy part of it by, say, for example, I just said content of galactose and content of fructose, there are other variants. Say, for example, mannose 5. In the drug world, biological drug world, mannose 5 and mannose 6 variants, these are called high mannose variants. So the green circles are mannose that are present in the antibody carbohydrate chains. So whenever you have high mannose content, it can affect clearance of the antibody from the body. So these are very crucial glycostructural features, variance features, which can actually impact safety and efficacy of the molecule, and that is why they are very, very crucial. You take care of these variants while you are manufacturing the drug. Now we will shift gears how to manufacture such a large molecule. What is the process through which we can manufacture these large molecules drugs. So normally we start from a genetic sequence. I told you that these are actually proteins. So you proteins, you have amino acids and then corresponding nucleotide sequence also. So this is a nucleotide sequence of a monoclonal antibody. So you have this is a heavy chain. I just showed here the antibody molecules. So these is the heavy chain, these are the light chains. So this actually is a uh, tertiary structure representation. So if you go down to the genetic sequence of this antibody, you will find nucleotide base pairs are arranged like this. So this heavy chain, which is the nucleotide content number 1353 amino acid, which will code for amino acids of 451 numbers and a molecular size of around 49.6 kDa. Similarly, light chain will have 639 with an amino acid number of 213 and molecular weight of 23.4 kDa. So, if you multiply the total molecule, two heavy chains and two light chains, total molecule of the whole antibody molecule becomes 146 kDa. So, that is what I was referring as a 150 kilodalton monoclonal antibody molecule size, which is actually huge, large in size. Why we are taking this sequence? Because we actually need to create a clone. This clone is the starting material in the manufacturing process. So first we need to create a clone, and to create a clone you need this genetic sequence, exactly producing your antibody molecule, coding for the amino acids, which will actually make your antibody molecule as a protein. So normally you take a vector like this, which is a DHFR based system, and this DHFR based system is DHFR is the hydrofolate reductase, an essential material unit for purine and pyrimidine, which produces a cofactor for thiamidylate synthetase. Now, this DHFR system helps you to select a particular clone against the selection pressure. You need to select the clone where you are putting this gene into this clone, and you need to get the clone in the expression host so that you have a stable supply which will go to the bioreactor or where you will need manufacturing in a large vessel. These cells are actually cultured. So genes get into this clone, and this clone then transferred to an expression host, mostly a mammalian process. When you want to produce large molecule drugs like antibody molecules, I have just shown you that you need post-translational modification of glycosylation in your molecule, and you need a mammalian cell because microbial cells, which can also be used for manufacturing biologics, cannot have this post-translational modification. It's a key distinction between mammalian cell and prokaryotic cells. So you need mammalian cells actually to produce this kind of molecules. So the industry workhorse of producing large molecules which has carbohydrate structures are Chinese hamster ovarian cells. So whatever clone you make with the genes of the antibody molecule put into the clone, you put that clone or construct, vector construct, plasmid vector construct into these cells. And then you select those cells which has the proper construct with the desired gene of the antibody molecule. So, and it is a long selection process through which you select those clones, cell lines, and make them stable, so that whenever you want to use it, you can actually thaw a vial and can put it in a culture vessel and can get the desired protein. So those are called cell banks. We normally in the industry call these as a master cell bank and working cell bank. Master cell banks are cell banks where the cells are frozen and kept in under liquid nitrogen. The temperature is around -196 degrees Celsius and kept it for a long term. Remember, you have to keep this manufacturing this drug molecule as long as the product is being used across the globe. So you need these vials also, frozen vials, till the time your product will be available in the market. So long-term storage is done for mammalian cells under liquid nitrogen, and you can thaw these vials and can put in a safe flask, revive the cell, and can start producing the antibody molecule, protein molecule from these cells. Cell line. Now it should be referred as a cell line because this cell, particular cells, now harboring the antibody genes, and it is making that protein inside the cells. Then you need to purify the protein from the culture and get your drug. The complexity of selecting a clone is being represented here. So here you have antibody levels, production level in the culture of those cells, and it's the probability of getting particular value, milligram per liter, volumetric productivity. Now you see, most of the population lies between 50 to 100. Few are there which are around 150. Now, this is very heterogeneous culture, and there will be, as you can see in the cell line, where most of the cells will be producing very little amount of your protein, desired protein. Very few will be there, as it is shown here, which will be produced which will have a production value, volumetric productivity value, which you can take it forward for a higher level culture, higher volume culture. So 90% transferrin produce less than 91 milligram, and 1.5% transferrin produce more than 150 milligram per liter. So you have to select this 1.5% out of total population. That is the challenge for a cell line to develop, which can be used for manufacturing. This is the challenge. So once you have those vials, you cell line selected, you frozen vials, you kept MCBs, master cell bank, and working cell bank under proper storage conditions, you know you have to go for manufacturing. So this is the outline, very simplified outline of a manufacturing process of a monoclonal antibody therapeutics. So it starts with revival of cell bank, and you revive that in a 125 ml culture. Then you go to safe flask level at 500 ml. Then you go to bioreactor at 5 liter level. Then you go to a bioreactor 50 liter level. Culture volume, these are culture volumes. Finally, this 50 liter culture of your cell line will be an inocular for the 500 liter bioreactor production bioreactor. And then the whole process starts after culture period, say for 2 weeks, 14 days or so. There could be culture time for 10 days or maybe 16 days. But generally, it is around 2 weeks. You harvest your culture when your cell viability in the culture is greater than 50%. You do a clarification step whereby you take out the cell debris, and then you reach to a critical intermediate stage. Critical intermediate before you go to make the drug substance and drug product, and you store that at a minus 20 degrees centigrade. So you have started with a vial like this at n minus 4 125 ml flask level. You have come to a critical intermediate, which is actually contains your protein, desired protein, and you store that at 20 minus 20 degrees centigrade. Then you have to purify. So this process, the left hand panel, actually the process we call it as an upstream cell culture process, upstream processes. So, or USP. Now, next is you have to actually purify this. This critical intermediate will have a lot of impurities and many other things which needs to be purified. You need your pure drug intermediate or drug API to be around 90, more than 90% pure. So how you get the purity? Well, you have a series of purification steps. You have the clarified bond, and because it is an antibody molecule, remember we have started with an antibody gene. You can use a protein chromatography, which will actually purify the protein to a level of more than 75 to 80%. Then you need to do low pH treatment, and remember your composition on your um, uh, drug formulation cannot have viruses and other pathogenic substances. So low pH treatment and neutralization steps actually takes care of that, and there are other steps which also takes care of that. But this one is the most key component. Then you do multimodal chromatography. Then you do tangential flow filtration, concentration and diaphragm. You need to concentrate, and then you put nanofiltration. Here again, some kind of virus removal happens. Then you add excipients and make the drug substance. This is all. This is not yet the drug. This is not yet the product which can go to the market. This is a drug substance, and you store that drug substance. Mainly, you purify protein, purify to the level of more than 90%, is stored at minus 20. Then you have to actually this step is called as downstream processing, purification processes. So downstream, so upstream, this is downstream to upstream, so downstream processing, and then you have to actually a fill finish stage where you do mixing of drug substance with buffer, and then you do sterile filtration, and then you fill in particular vial or PFS, pre-filled syringes, where you know your target product profile should be TPP. If the drug, you are lyophilizing the drug, it may go into the vial, and or if it is a solution, you create a pre-filled syringes where the drug is actually loaded into the syringes. So all these things happen, filling is happen, then you do visual inspection and labeling and storage of vials at 2 to 8 degrees. This is ready to go to the market. Now, I have shown here all the equipment that are gone into such kind of manufacturing processes. This is the bioreactor. This is 6 plus. This is bioreactors, bag. Even in bags, these bags, rocking bags, you can culture cells and produce proteins. And this is also bioreactor, and these are purification setup where you, for column chromatography, you put your matrix, resins or matrix into these glass cylindrical tubes or vessels, and you do purification. And then this is a lyophilization and a fill finish machine as well. So this is a very simplistic overview of a manufacturing process of an antibody therapeutics. And this process, I told you, this culture stages takes almost 2 weeks of time. Before that, you have to propagate the cells from 500 liter. Here, it is almost 2 weeks, and the whole process can actually take 3 to 4 weeks, where you get finished product to go to the market. So you have a product now, but because by being it's a very large molecule, complex molecule, lot of structural features, molecular structural features, which needs to be characterized. And these things happens in the even before you take the molecule to the manufacturing stage. You need to know its primary structure, determine higher order structure, because these are protein molecules. Then its biological function to be re-established or established, and then product-related substances and impurities. These are all product-related information. And then impurities you have to actually also study, because if you have impurities, that will be a safety concern. That can be a stability concern for these drugs. So you need to actually test for general properties and excipients, process-related impurities. You need to test by various orthogonal methods. Then you need to check also particles and aggregates, because aggregates, if your proteins has a natural tendency to aggregate, and you are actually concentrating the protein to put it in a vial and all. So aggregates in protein, and if that goes to the human body, we know proteins, aggregated proteins at a very high concentration will generate huge immunogenicity. That again is a safety concern. So particles and aggregates also needs to be checked and appropriately formulated for the drug. So this is called the process flow for manufacturing of biological drugs. You start with cell line and close selection, then you've done upstream, then you go to downstream purification stage, upstream where you actually culture the cells, and then formulation and drug substance and drug product. And all of this analysis you need to do at all of these stages. So we call that as a critical quality parameters in case of biological drugs. These critical quality parameters, because it is critical, why it is critical? It can significantly impact the two key component of the drug's action, efficacy and safety. So I have told about immunogenicity. I have told about efficacy, how it is being impacted by galactose content and fructose content. I have told about mannose, role of mannose, high mannose variants in molecular structures, how it can actually control clearance of the molecule, and if there are impurities, of course, they can again contribute to immunogenicity, means safety and stability. So all these critical quality parameters are listed. Whatever is listed need to be studied through and through all these stages in a drug manufacturing processes. So you have made this. This is a graph which shows how over the years cost per gram of this antibody therapeutics or any biological drugs have been reduced because of the advancement made in the manufacturing processes and bringing automation to the manufacturing processes. So pre-1995 it was around $500 to $1,000 per gram of this antibody or any biological drugs, around 500 to 1,000, and then you come to future. Around 15 to 25 is around 22. Currently it is around 20 to 50. So some guys who have actually implemented continuous manufacturing processes have reported that they have reduced it to around $40 per gram of protein, per gram of the drug. So it's a huge reduction over the years, and in future people are actually targeting to reduce it to 5 to 10 grams, $10 per gram. It's an interesting information. So where do we stand in India as far as biopharmaceutical is concerned? Let us look for the values that have been created from the biopharmaceuticals. So this slide shows a pie chart of Indian bioeconomy in terms of the biotech sectors, and you see biopharmaceuticals could include vaccine, therapeutics, diagnostics, and this contributed around $16.8 billion for vaccine, $14 billion for therapeutics, and $19 billion for diagnostics. So it's a huge jump. And then rest of the biotech sector also contributed. So the total bioeconomy of India is around $137 billion, which is roughly 2% of GDP. And I referred earlier that the total pharmaceuticals in the world, the value is around $1.9 trillion. The pharmaceutical world, drug world, and including vaccines. Here, biopharmaceuticals in that is around $665 billion. So around 35% of the total pharmaceutical world is actually biopharmaceutical, and it is growing at a good CAGR. So because there are plenty, I mean, there are significant benefits as far as target specificity is concerned, as far as manufacturing complexity is there, but it is not considering the trade benefit between its target efficacy. I mean, efficacy because of the specificity, targeted specificity, its efficacy is also ensured, and we can actually have better relapse-free survival in cancer because of this development of antibody therapeutics. And there are many other development is going on, which we will come back and we'll discuss later. And so biopharmaceuticals, and we all know about vaccines development during COVID-19. So this biopharmaceutical world is growing at a huge pace, and we should track this area, this particular segment of pharmaceuticals, because in future already FDA approvals are 50% at 50%, and most of the experts suggest that in another 10 years, biopharmaceuticals will be 50% of this total pharmaceuticals world. So traditional pharma, small molecule drugs, which will be halved by the biopharmaceutical drugs. I hope this is very small, I mean, little information. I wanted little significant information I wanted to impart through this lecture as an introduction to the manufacturing of biologics. Thank you very much.